All right, so today we're going to finish chapter 6. We just did a little introduction to it last time. So today we'll finish chapter 6, and then your test will be over chapters 1 through 6. And it will be pretty evenly distributed between all of them. It will be all multiple choice, matching, and true false, so you don't have to type anything out on this test. And then that's it, okay? All right, so anyway, chapter 6, viruses. So in this chapter, we're going to talk about how viruses, and I'm not going to mention a whole lot about this actually, but the early discovery of viruses was basically that we knew that there was something else causing diseases, and it wasn't bacteria, because we could see bacteria under a microscope. So it had to have been something that we couldn't see yet. But then once we designed electron microscopes, then we could see what those were, the viruses. We're going to draw the structure and label the components of enveloped and non-enveloped viruses, and then describe the 5 stages of viral replication, and explain 2 ways that emerging viruses might arise. There are others, but we'll talk about the 2 main ways. Describe how viruses can cause tumors and therefore cancer. And then we'll discuss the truly creepy, the viroids and the prions that aren't even actually full viruses, although they can kind of act like them, and they're even more simplistic than the viruses are. Okay, so the virus structure has, before we even talk about the structure, let's talk about this obligate intracellular parasite issue. And I know we mentioned this last time too, but it's worth re-mentioning. An obligate intracellular parasite means that it is a parasite, which means it lives off of another thing. Intracellular means it lives inside of our own cells. And obligate means that it must do that. So it has no other option other than to, quote, live inside of our cells and other cells like plants, animals, bacteria. All of those things can have viruses inside of them. Now, remember, the reason I said live, and I'm trying to do vocal quotes there, but is because they are not alive. That's one of the very important things you need to understand about viruses. They are not living. They do not metabolize. They do not reproduce themselves. They can't move. So they don't follow any of those criteria that we consider for things to be living, okay? So it's the host cell that is going to reproduce the virus. So the host cell gets fooled into taking in the virus and then replicates it like crazy and spits out the results, the finished viruses, okay? So this picture is showing you some of the different sizes of, a cell like ours on the far right. That eukaryotic cell could be like our size. At the bottom is a bacteria that we've already talked about. And then all the rest there are viruses, and so they can be about as big as a bacteria all the way down to the teeny tiny little parvoviruses and other very, very small viruses. So a large variety of sizes, but measured in nanometers, which is the next step down after micrometers, which is the next step down after millimeters, or nanometer, millimeters, sorry. And then this next step down from that one is the centimeters that we're used to talking about. So, like 4 degrees down, so quite a bit of difference in magnitude of size there. Okay, so as I mentioned last time, we have viruses with 2 components that they absolutely must have. Their genetics, which are the DNA or the RNA, but never both, and then capsids, the protein coat. So they all will have those 2 things. And it can be a pretty fancy looking coat, or it can be a very simplistic coat. The DNA can vary in length, same with the RNA, but again, they don't have both, it's one or the other. So we can kind of categorize viruses based on, do they have DNA or RNA, and that's a pretty good way to distinguish them. What we can see here are some of the different shapes of the capsids. Okay, they're protein coats. And they're showing you here, give my little pointer. This red swirly capsid is called a helical protein coat. And it is just like a a really tightly wound slinky, kind of. The next one then is this fancy little geometric design one. It's called icosahedral, and it has just multiple sides, and it's kind of crystal looking. And then they can be complex. So this one obviously has an icosahedral top, but then some more appendages off on the bottom. And so they do come in very different types of complex structures. And then some of them also will have the envelope, which is the outer layer that is more of like a plasma membrane covering that our own host cells will put onto that cover, and it may have spikes attached to it then. And the coronavirus is one of these types. So the coronavirus has RNA and a protein coat and a viral envelope, very similar to this one on the right side. Okay, so now we're going to look at how our cells replicate a virus. Okay, so this is taking you through those multiple steps: attachment, penetration, biosynthesis, assembly, and release. Okay, those 5 steps. And the next page will show you a picture of that, but let's kind of look through these on here as well. First off, you can see, before I go too much further, that little latency orange box off to the right. That one is when a virus will get into us and then just sit there. We'll talk about that in a little while. I just wanted to get that out of your mind for a second. So attachment is when the virus is attaching to the host cell. And it's not, the viruses cannot swim up to a cell and say, this looks like a good place for me to infect, I'm going to hold on to this. They are simply just molecules. They are only DNA or RNA with a protein coat. They can't produce energy. There are no neurons there to think with. So it's literally just that they bump into a cell, and the cell then has little receptors on it that they can take advantage of. So the cell is really the thinking part here, and the host cell, which is in us or in plants or in bacteria, recognizes the virus and says, hey, this is something I should take in. It just tricks it, okay? We've evolved together, and our cells have been tricked into taking these viruses in. So attachment is just the virus holding on to the cell. Penetration then is when some portion of the virus gets into the cell. And the reason I say some portion is because in some viruses, a large part of it will get into the cell. In some viruses, only the genetic information gets into the cell, okay? So the nucleocapsid may enter the cytoplasm, and then the DNA gets out, okay? Or just the DNA gets injected. But regardless, now the viral genetics are inside of the cell. The third step then is biosynthesis, where it's just what it sounds like, life development, okay? The viruses aren't alive, but anyway, we are synthesizing the viral genome, which means the DNA or the RNA, as well as the capsid, so we make a new protein coat, and any spikes that are needed as well. So where does this information come from? How does our cell know that this is what it should do? It's in that genetic code. So the genome programs the cell to do what it wants. So basically, its mere presence in there makes the cell make more of itself, okay? So it would be like, if a loaf of bread shows up in your house and you're like, oh, a loaf of bread, I should make more loaves of bread, right? The loaf of bread isn't living, but I'm the living part here, and I'm going to decide to make it, okay? So that's kind of how it works. Like the DNA has the genetic information to tell the cell, make more genetic information just like this, and here's another gene for making my protein coat, and here's another gene for making the spikes. Okay, so our own cells then, the machinery of our own cells that should be, making its own molecules, is now going to make all these viral molecules. All right, and then those all have to assemble. So that's our fourth step, assembly. The capsid proteins are going to be wrapped around and then the envelope is going to form on release, okay? So that next step is release, and that's when we have the nucleocapsid, which means the genetic information inside of the capsid, bumping into the surface, and the spikes can be on that plasma membrane already, but basically you're going to wrap that capsid in the cell's own membrane and stick the spikes on there, and then it's pushed out of the cell. Okay, if that happens nice and slowly, then the cell might be able to recover from that. Yes, it will punch little holes in the cell, but the cell can quickly close those holes up. And then those viruses can go on to infect other cells. What happens sometimes, though, is that so many viruses are leaving the cell that it essentially explodes with viruses. And there are too many viruses leaving for the cell to be able to fix that plasma membrane. And so it's just like if you were trying to, force a whole bunch of tiny little balls through a bubble. Eventually the bubble's going to pop. Okay, it just can't reform itself. So here's a picture of the same thing. And if we just follow this up here as stage 1, where we can see in this example both the DNA and the capsid get into the cell. Then the DNA is released from the capsid and makes its way into the nucleus. And within the nucleus then, the nucleus of the host cell is going to make a whole bunch of new pieces of DNA and a whole bunch of new spikes and a whole bunch of new capsids. And in this case, it's attaching those spikes to the nuclear envelope. So this is the plasma membrane that is surrounding the nucleus right here. And so it attaches these little spikes to the nuclear envelope. And then we have the DNA with a capsid with an envelope wrapped up in this little protein membrane, phospholipid membrane, excuse me. And then that is moved out of the cell through exocytosis, which is a process of just moving big things out of the cell. Okay? So then you can see that we've done all 5 of those steps, and now these viruses are just like the ones that came into the cell, except that we're making hundreds, thousands, millions of them and pushing them out of the cell. Okay, this picture is showing a little bit of how just the genetics can be inserted into the cell. So you can see that the virus is here, and it attaches to that little yellow protein that's on the cell. The host cell is at the bottom of the screen. And then in the it opens a door, basically. The host cell kind of opens up the virus and allows the genetics to come in. And that's what we see here now, is that the genetics of the virus are inside of our own cell. Okay, so this one is just focusing on penetration for this picture. Any questions on that? So this is one of the most important things from this chapter, and make sure you study that information. Okay, so I mentioned that little orange box a couple slides ago and latency. I want to make sure that you understand what latency does. Latency is a way for the virus to stay in you and just resting until you are stressed out, and then it can come out and do its nasty work again. So chickenpox is an example. So what happens with this varicella zoster virus is that's what we know as chickenpox. So, a lot of the students in this class are younger, so I'm sure most likely you've not had chickenpox. But anyway, the way that this works is you get the virus, and then you break out in red itchy bumps, and then the virus goes to hide in some of your neurons, which are your nervous cells. And those neurons will just house it. They just kind of sit in there and rest, because the DNA, the virus is just sitting in there and waiting for you to be stressed out. So usually sometime after you're 40 or 50, that virus comes back out when you're stressed, and it comes out as shingles, only along those neurons where the virus had been hiding. So usually along your trunk somewhere is where the virus will come back out of those neurons and cause intense pain. HIV is even trickier. HIV goes latent by becoming part of your own genetic code. And I think I mentioned this last time, but the little red viral DNA here, just this red section, gets inserted into your own DNA strand. And that's really horrible, because then to try to disable HIV, we've got to try to get that DNA code out or shut it down or something like that. But anyway, so you can have HIV for years and years and years, having that human immunodeficiency virus, but it only turns into AIDS once it comes back out from its period of latency, okay? And then it's acquired immunodeficiency syndrome. That's what those stand for. Okay, so once it comes back out, then it can be very bad, because then it's compromising your immune system all over the place, and you end up actually not dying from the AIDS. You end up dying usually from some other infection, because your body can't fight it off. Okay, so that's a pretty important concept to understand when we're talking about viruses, is this ability for some of them to go latent. And one of the scary things about coronavirus is that we don't know yet if it has any ability to be latent. So far, we have seen lasting damage from coronavirus, but we have not yet been able to pinpoint if it goes latent or not, because we haven't had enough time. Okay, oftentimes, with shingles, it doesn't come back for decades. And you can, if you properly manage HIV, it can stay latent for decades. So we don't know yet if this COVID-19 has this ability, and that's why you should not, absolutely should not, assume I should just get COVID and then be over with it. That is not how this virus works. That is 100% false. Okay, there are lasting effects from COVID-19. We see that for sure, because it affects your blood vessels. Therefore, it can also affect your lungs. And so you can have lasting effects on your heart or your lungs or your kidneys. But we don't even know yet if it can go latent. That's a totally separate issue, and we have no answer for that yet. Okay, so one of the ways that our body can fight against this replication is through the immune system. Okay, and bacteria can do this too. I'm not going to get into how bacteria can protect itself, but there are definitely plenty of viruses that can infect bacteria, and viruses are going to try to stop the replication of the virus as well. But in humans, we have our immune systems, and there are these three main ways that our immune system can fight off viruses. Number one is that we have antibodies that can cap a virus. Okay, so I'm going to just draw a little picture here. So assume that this is a virus, and it's got these little spikes on it. Okay, and these little spikes are a type of antigen, which means that they are a way that our immune system can recognize them as belonging to us or not. Okay, so you hope that a virus displays these antigens and that our immune system can recognize that they are not of us, that they're some foreign thing. And then you can have these, your body will make antibodies, and they typically look like a little upside down Y. You can draw them like a Y, but they're kind of represented like that. And the antibodies are produced by our own cells in response to what is in the body. And they will just cap a little virus like this. Okay? And then when that happens, that will really reduce the amount of viruses that can get into the cells, because oftentimes they'll use those spikes as a way to get into our cells, to kind of trick our cells into taking them in. So if we can stop that, if we can kind of stick them up with all these antibodies, they can't get into the cell. Okay? Now, if you have too many viruses in you, so let's say that somebody, for example, sneezes or coughs in your face with COVID-19. Enough viruses could get into you that even if your body is able to produce antibodies quickly, there may simply be too many viruses in you to be able to react to that. And also, it takes your body a little bit of time to make the antibodies anyway. It does have some that are kind of generic and can kind of cap anything foreign, But if it's a novel virus that your body has never seen before, then those antibodies may not be working, okay? Or they may not be prepared yet because your body is just having exposure to this virus, okay? So that's a couple of reasons why even if you do have a healthy immune system, you can get COVID because you've just received enough of the virus in you that your body can't fight it off quickly enough, okay? All it has to do is get into a few cells and start replicating for you to be sick from the virus. Okay, another one then is the immune cells. Okay, immune cells are a little bit more, are very more complex actually, but immune cells have ways of attacking foreign objects and destroying them. So you hope that you have a healthy immune system, you know, things like natural killer cells, T cells. Those are going to attack a virus when they find it and try to disable it. And so they can actually, if I draw, I'll stick with my color scheme here, and I'm just going to draw a much smaller virus compared to the size of our cell that could attack it. So I'm going to draw the cell that is ours attacking it like this, and it can come and literally eat up that, there's the nucleus, now it looks like a dinosaur, can eat up that little virus and hopefully disable it. So we do have cells that can do that. And again, you hope that you have enough of these cells to keep you from getting too sick. That's one method our body has. The last one then is just a molecule. Interferon is just a molecule that has the ability to disable viruses. And again, we're not going to get into how it does that, but it's your body's natural way of fighting generic viruses. Any virus it sees, it's going to release interferon, which is going to attempt to destroy the virus. Okay, and probably nobody in here has seen this. This was a movie that came out, I don't know, must have been in the 70s or 80s. 80s or, yeah, probably the 80s. But it was called Fantastic Voyage, and the gist of the story is that they shrink these people, doctors, into this teeny tiny, you know, microscopic size and put them inside of this guy's brain because he's got like a brain tumor. So they shrink the people, put them into the bloodstream, and they're going all throughout the body, like driving this little yellow submarine through the blood vessels and everything, trying to get up to the brain so they can get rid of this brain tumor. And what you see in the bottom picture is that one of them gets exposed to antibodies and gets covered in antibodies because the body recognizes that this miniature person inside of an actual person is foreign. But it's kind of a terrifying image because all these little like jelly-like creature things, which are the antibodies, come and try to attack her. And they do, and they kind of like suffocate her. And so they're in this video, in this picture here, they're trying to tear all those antibodies off of her to save her life. So I think that had an impact on me. If you like cheesy 80s movies and science, that's a great one to watch. All right, anyway, so let's talk about viral diseases that are emerging and why this is such a big problem. And this is exactly what COVID is, okay? So emerging viruses are viruses that are new to humans or just new to an area. Okay, we can say, you know, for example, Ebola is one that had been in other parts of the world, but hit West Africa pretty heavy in 2014. Okay, so anyway, emerging viruses are new to humans or new to an area, and they can become endemic, meaning that they stay in that area. They weren't historically found there, and it might be just a flare-up and then it goes away, but some of them may stick around and be in that area for the foreseeable future. Okay, so here are some examples, and you have certainly heard of these before. So H1N1, that H1N1 flu that spiked up in 2009 and had everybody worried, but then we got it under control because it wasn't as contagious as we feared. Polio made a little flare-up in 2014, and we tamped it back down. Ebola in West Africa in 2014, and we still see Ebola pop up every now and then in some weird places. Zika virus in 2016 in the Americas, North and Central and South America. So we see these, and then of course right now we have COVID-19 as our big problem, really big problem. And we do call it a pandemic because it is worldwide. And it is new to science. So it was one of these examples that we see at the top, the zoonotic at least we're pretty darn sure it was, where it was in an animal and then it jumped species and is able to infect humans now. Viruses can be extremely specific. So there are viruses that only affect plants, and there are viruses that only affect certain species of mammals or certain species of reptiles. They can be very specific. So oftentimes there's some event, some change that allows them to now infect a whole other species. Okay, and sometimes it's surprising. It might be something that's only found in birds and then it hops to pigs or to humans. So some of these zoonotic spreads can be surprising. And the big problem with these novel viruses, which means they're just new to science, is that our bodies are not prepared for them. Okay, our bodies have no natural immunity to them, and so we are all then potentially infectable. Okay, so it does not seem that there is anybody who is guaranteed to fight this off. Okay, there are certainly people who are more susceptible to it, and it definitely appears that the amount of the virus that you're exposed to will predict how sick you become from it. There are some strong pieces of evidence there. And of course, we know once it gets in you, it can have various effects on your body. But the fact that COVID-19 is more contagious than a lot of other viruses is a huge cause for concern, because then it can move very quickly through a population, especially through the normal means that we all think, as long as you're not, with some viruses, as long as you're not exchanging saliva, they're very difficult to pass from one person to another. Okay, it would be fairly easy to go to a foreign country and avoid a lot of their endemic viruses simply because you have to have that kind of contact with another person to be able to get the virus. It's just not the case with COVID-19 because it travels through respiratory droplets. All right, so they are predicting that we will continue to see these types of virus outbreaks, and viruses are so much harder to control than bacteria because we can't watch them in the same way. It's harder to test for them in some cases. It's also harder to follow them through your cells, find them in your body because we can't look at them just on a light microscope. It's also more difficult to, what was the last one I was going to say? Oh, to tell if they are still contagious. That's a really big one. So for example, they keep refining the science on how long the virus can remain viable on surfaces. Because with bacteria, you can just swab a surface and grow it on the proper petri dish. And if it grows, it's still alive. Well, you can't do that with viruses. You can't put a virus on a petri dish and have it grow. It needs host cells. Okay? So we'd have to get the virus onto host cells and have the host cells be living and able to replicate viruses. And then we would have to look at those cells to see, oh yes, they do show viral replication. Okay, so that whole process is just way more difficult with viruses. All right. All right, any questions on that? Oh, I do want to mention that climate change is making it worse, and we're having more trouble controlling the spread of viruses as climate change warms up climates. So one example is the spread of mosquitoes. Okay, if mosquitoes carry certain viral diseases, which they can, as the Earth warms up, those mosquitoes can travel farther and farther away from the equator, and so we will see more of those occurring. Also, the other things that can make emerging diseases more likely are international travel, the shipping of goods, overcrowding and pollution, having people be more in contact with organisms that they not have been before. Okay, there's one other way that we can have new viral diseases emerge, and that is what you can simply call just jumping viruses. No, they don't have little legs to jump with. It's not that kind of jumping. What we mean is their genetics can jump. from one virus to another. Okay, so we can have a little piece of viral DNA from one virus in your cell, for example, and if another virus is also there, then it kind of can mix. And then when your body will package those capsids for release, some of the genetics of that second virus could get into the capsid of the first. Okay, does that make sense? So you might have, a cell and two different viruses get in. So we'll do like a little red virus and a little purple virus. And let's say that they both get in, and as the cell is remaking their genetics, then they will get mixed up. So you might end up with a virus out here that has some of its own genetics, but then some of the purple virus's genetics in it as well. Okay? And so you will have then maybe a virus that didn't have an envelope before now has an envelope. And those envelopes can, one second. Sorry about that. So you can have a virus that didn't have an envelope before that now can develop an envelope, which can mean that it's more protected than it was before. Okay, or you may have one that has spikes, but the new DNA gives them more spikes. Okay, and that can make them then unrecognizable, even though a person had immunity to the first one. When they get that new mutation, they might not have immunity to the second one with more spikes. Okay? All right. So now this is a really interesting topic. This is looking at the role of viruses in causing cancer. And you might already know one of them with the human papillomavirus being able to cause cervical cancer. And so that's one that, students your age should typically know about, but it can happen with many different viruses and cancer types. So some students don't really understand what cancer is. So let's go through a real quick review of that. So if you look at the notes for this section, which are posted in Blackboard, I do have all this written down, so you don't have to take too many notes here. But cancer has several attributes. It has uncontrolled growth, which means that the cancer cells divide like crazy, and it does not have an automatic kill switch. So some cells, all cells really, have this programmed what's called apoptosis. It looks like apoptosis. But apoptosis is programmed cell death, so that if a cell is behaving weirdly, it will self-destruct. Unfortunately, cancer cells, that gets turned off. So they divide like crazy and make more of themselves. They don't die when they should because they're acting weird. And then the third one is that they are kind of just stupid cells. They don't act like they should. They're just big eating cells. They're just greedy, and all they care about is eating more food and growing more of themselves. So as an example, if you have liver cancer, the cancerous cells are not doing what the liver should do. Livers are there to detoxify molecules and process all of your food. And if you have liver cancer, those cells are there, but they're not doing that job. They're only eating and making more of themselves. That's it. Okay, so again, cancer has uncontrolled growth. It doesn't die when it's supposed to. The cells are stupid and they just eat all day. And then the last thing is that they don't stay still. Okay, other cells, normal cells will be bound to each other better, but cancer cells are okay expanding and breaking off, and that's when they become metastasized. So metastasized means that they have escaped where they were, and they are now roaming around your body, and wherever they land, they can start growing again. Okay, so tumors are just areas of accelerated cell growth. So if it's a benign tumor, the cell is still rapidly dividing, but it hasn't broken out of its little capsule. Okay, the body is able to kind of keep it in that space. If the tumor is malignant, then it is rapidly dividing too much, and those cells are cancerous. and then they will shortly after metastasize if the body can't keep it under control. Okay. You probably have a few cells inside of you that are cancerous now, but your immune system can keep those in check. A cancerous cell is just one that has gone haywire. Okay. But if that happens too much, or if your immune system can't keep it under control, then the cancer will grow to a noticeable level. Okay. So oncology is the study of tumors and cancer. Just as this, if you've heard of that field before. And that term oncology gives us a clue to this next slide. And this is talking about how viruses can be involved with the creation of cancer. Now, first off, we do have materials that are called carcinogens that are just things that cause cancer. Carcinogens are anything that causes cancer. So that could be like ultraviolet light, radiation from a nuclear power plant, certain chemicals that are, if you ever see those little stickers for known to cause cancer. Well, viruses are one of those too. So viruses are considered a carcinogen. All of these things here, these are all carcinogens, okay? And so what happens is they take a normal cell and alter the DNA so that it just starts acting abnormal and doing all those things that we already talked about, okay? A lot of cancers can just be caused by mutations of the body, just mistakes when the cell was dividing and something happens to the genetic code, okay? But we know that there are certain things that cause cancers, and viruses are one of them. If we know that the cancer was caused by a virus, we call it an oncogenic virus, okay? And therefore, human papillomavirus would be called an oncogenic virus because it has the ability to induce cancer in those cells where it has done its work. Okay, so for example, cervical cancer. Okay, and it does this, I don't need you to know the details of this screen, but I do need you to know that the virus literally is messing with your genetics when this is occurring. That's the only way that we can have a cancerous cell, is if the genetics are in error. Okay, we've made a mutation to those genetics. So basically, the viral genetics inhibit a tumor suppressor gene. So basically, you have a gene inside of you that prevents tumors from forming. Okay, prevents them from dividing too rapidly, and prevents them from not dying. I know that's a whole lot of double negatives, but basically that tumor suppressor gene is there specifically to stop tumors. So your genetics naturally have that. The virus comes in and slows that down, inhibits it, stops it from working. So now if you have a cell that is going to form a tumor, there's nothing to stop it. Okay, the virus has stopped the ability of your cell to stop the tumor. Okay? All right, so that's it for tumors. Now, I'm sorry, for viruses and tumors for that matter. But now we're going to switch from viruses to viroids and prions. Okay? So viroids are really freaky because they are like a virus in that they have, in this case, just RNA with no protein coat. So they are just a strand of RNA. That's literally it. But you can see on this one that it is circular. And so basically, it's a very strange material. But if I was following the strand of RNA, I'd go like this. This is one side of the RNA, and then it kind of loops back on itself back this way. And then these little lines here are where the nitrogenous bases would be. So it's a weird thing. It's just this little circular piece of RNA with, A's, U's, C's, and G's, adenine, uracil, guanine, cytosine. And that's it. It doesn't have any cover on it. It seems like it would be, harmless and easy to destroy. but because it doesn't have any protection, it's kind of protected in its simplicity. So anyway, it's viroids aren't too much of a problem to us. They all, except the fact that they can cause crop diseases because they only infect plants. Okay, so viroids only infect plants and can cause some crop diseases that are harmful. That's really their only effect on us. It's the prions you really have to watch out for. So prions are only proteins. So they're kind of like, they're not even the protein coat of a virus. They're just proteins. But they are proteins that, again, our body will replicate. And the prions can and do infect animals, including us. And they cause neurodegenerative diseases, meaning basically they destroy your brain cells. So mad cow disease is an example. And then this Creutzfeldt-Jakob disease, there's a type of that is caused by that is caused by prions that can infect us. And so what happens if you have a prion is that it basically just slowly destroys your brain tissue until it looks like Swiss cheese. And the creepy thing is that these things are extremely difficult to get rid of. And so this becomes a really big problem during surgery. So let's say that you have surgery, and you had surgery after somebody who had prions. So somebody else got prions in one way or another. And then the doctor will autoclave the surgical equipment, the scalpels and everything. And when you autoclave, that kills like basically everything, even bacterial endospores that are almost impossible to kill. It kills those too. But it cannot get rid of prions, which is crazy because it's just a protein and we should be able to denature those, which means cause them to lose shape and functionality. But it doesn't work. We have no way of getting rid of prions off of surgical equipment. So if you then have brain surgery and they use the same equipment, then the prions will be in your brain and your brain cells will start manufacturing more of them and your brain will end up as Swiss cheese. So they are catastrophic, and it's not too often that this happens in surgery, but it makes the news when it does. So if you start paying attention to medical news articles, you'll see that, it'll be mentioned somebody had brain surgery, the previous person, they didn't know the previous person had prions. If they would have, they would have just thrown away the equipment. And so it doesn't guarantee that you're going to get the prions, but it's possible. All right, but for our day-to-day life, not too many prions for us to worry about, thankfully. Okay, so we learned a lot about viruses and how they replicate and how we can fight them off and how they unfortunately can lead to cancer formation. And we learned about prions, which affect us, and viroids, which affect plants. Okay,